My name is Reidun Twarock. I'm Professor of Mathematical Virology at the University of York in the United Kingdom. I'm a mathematician who is interested in the structure of viruses and their consequences for how viruses form, evolve, and infect their hosts. I will take you on a journey about virus structure seen through a mathematical microscope. A fascinating property of viruses is their high degree of symmetry. The majority of viruses have icosahedral symmetry, which means that they are sharing their symmetry properties with an icosahedral reference frame. They have 5-fold symmetry axis through opposite vertices, 2-fold symmetry axis through the centers or opposite edges, and 3-fold symmetry axis through the centers of opposite faces. However, icosahedral symmetry does not fully constrain virus architecture, as you can see in the examples in the middle. All these viruses are icosahedral viruses, but they look very different. So the central question for the mathematician is, what are the mathematical rules that account for these different incarnations of icosahedral symmetry? Crick and Watson proposed an argument for why viruses use symmetry. They reason that by using multiple copies of identical protein building blocks to build the capsid, the virus only requires a relatively short genetic sequence to code for the container, i.e. it codes for those building blocks that are then repeatedly synthesized and used for the construction. Caspar and Klug followed up on this argument and asked what could be the building principles of these containers. They were inspired by viruses like the one shown on the left that is formed from multiple groups of 6 proteins and 12 groups of 5 proteins, which are required according to Euler's theorem in order to get a closed shell. They embedded the icosahedral surface, the surface of the icosahedral reference frame, in the plane such that the vertices are located at the midpoints of hexagons on a hexagonal grid. They then investigated all options of achieving this by rescaling and rotating the reference frame. Each of them can be used to construct a polyhedral shell that is given in terms of 12 pentagons and otherwise hexagons, like those shown on the right-hand side. These form the start of the so-called T-number series, a famous series in virology that is used to describe viral architectures. The construction of the T-number series has been covered in our teacher packs that you're very welcome to download from my website. The link is given below using the teaching tab. We would be very grateful for any feedback. There's a form at the bottom of that page. If you enjoyed working with this material, have suggestions for things you would like to see, then please leave us a message on that feedback form and enjoy working with this material. I've worked on mathematical approaches to describe viral architecture for over 15 years. Early work was tiling models of viral surface, as you can see on the left-hand side. In particular, this example shown here, where I used tilings to explain the surface structures of the cancer-causing viruses that were long-standing open problem in virology in view of Caspar-Klug theory. Quite recently, I worked with a colleague from the US, Anthony Luke, on a generalized principle of virus architecture that encompasses Caspar-Klug theory, but also covers the many outliers to that theory that have previously not been covered in their framework. This mathematics has a lot of applications, for example in nanotechnology. People use proteins to create particles that are then used in vaccine design. An adaptation of viral tiling theory has been instrumental in understanding what type of particles can be formed from these basic building blocks. Viral tiling theory has also inspired work with artists. Early on, we worked on sculptures and projections, as you see at the bottom, together with summer students. But at the moment, we're working with a VR team, people experts in virtual reality, to render the tiling models in such frameworks for exhibitions at galleries, such as the Lottery in Manchester. If you want to learn more about that aspect of my work, there is a page in the Art and Science tab on my webpage that gives more information. Mathematical virology has acted as a driver of discovery in virology. A better understanding of the 3D structure of viruses has enabled us to understand constraints on how the genetic material is organized inside of these containers. This is known as the Hamiltonian path approach because these constraints are akin to the traveling salesman problems and constraints that, for instance, occur in the Gaussian game that you see on the right-hand side that Hamilton introduced. Using that type of mathematics, we've been able to discover a code that's hidden within the genetic code of viruses and guides how viruses form. Understanding this code is a step in developing novel forms of antiviral therapy. A recent article in Mathematics Today gives an accessible description of these mathematics. Given the COVID-19 crisis, we are of course all interested in what this virus looks like. Despite its spherical appearance from the outside, this is actually not an icosahedral virus akin to the ones we've been discussing so far. What you see is a lipid bilayer with the glycoproteins shown in red on the outside. These are not in icosahedral positions. The equivalent to the protein containers we've discussed before is actually here a helical structure shown on the right-hand side with the viral genome wound around its helical turns. We've made good progress so far in understanding the mathematical principles according to which it is built and in using this insight in order to better understand how this virus forms. I would like to thank my wonderful team who are working on different aspects of viruses, as well as my collaborators, in particular my experimental collaborator Peter Stockley, with whom I'm working on this topic for over 15 years and jointly have made important discoveries.